For many years, the standard treatment for patients with hormone receptor positive cancers has been the use of endocrine therapy. Now, in the last few decades, we've witnessed a shift from starting with chemotherapy for metastatic cases to starting with endocrine therapy, which of course has fewer toxicities. In fact, recent studies have shown that starting with chemotherapy or hormonal therapy gives you equivalent outcomes, maybe even slightly better outcomes with hormonal therapy, and certainly better quality of life. When aromatase inhibitors first came into clinical use in the 90s, they were compared to tamoxifen and were found to be roughly equivalent or maybe slightly better in some studies, but they had fewer side effects. For example, the clotting risk with tamoxifen wasn't seen, the risk of endometrial hyperplasia. So this became the standard for most patients, certainly for postmenopausal patients. And also, for premenopausal patients, blockade of ovarian function was important. That was actually described nearly 20 years ago, and that has become a standard, either ovarian blockade with tamoxifen, and then since the aromatase inhibitors, of course, using aromatase inhibitors. Now, in the last 4 years or so, we have now had the development of biological agents. In fact, everolimus was the first approved now 6 years ago in 2012 in the second-line therapy as we've started to understand the mechanisms of resistance, particularly activation of growth factor pathways, which are blocked by mTOR inhibitors. More recently, the cyclin-dependent kinase inhibitors came into the forefront with the first approval now 3 years ago. So the integration of these new therapies clearly has represented a benefit, but has left a lot of questions about what is the role of endocrine therapy alone and what is the best sequence of therapies to use. Initially, the development of cyclin-dependent kinase inhibitors was greatly improved when more selective inhibitors were brought into the clinic that had fewer toxicities, particularly the CDK4/6 inhibitors. The first of these to be tested was palbociclib, and the preclinical data suggested that the activity was seen more in hormone receptor positive cells, that these were more sensitive to these drugs, and that they did require endocrine therapy to be partnered with them. The first of these trials of PALOMA-1 study was a phase 2 study, a randomized phase 2, that also attempted to look at which biomarkers might be important. It was known that there are some aberrations in hormone receptor positive cancer cells in the cell cycle machinery, for example, amplification of cyclin D1 or loss of the retinoblastoma protein. And it turns out that in these early studies, there didn't seem to be an impact of these biomarkers. So the larger phase 3 study, PALOMA-2, was then activated comparing letrozole alone to letrozole plus palbociclib. And this was the first of many studies that have shown very similar results, and that is a doubling of progression-free survival from approximately 10 to 20 months to 20 to 24 months in that range, with pretty good toxicities, mostly with neutropenia that is subclinical, some degree of fatigue. And then, of course, the following other CDK4/6 inhibitors that were also in preclinical development ultimately generated the results from their pivotal studies, ribociclib being the next one, also very similar design, letrozole plus or minus ribociclib showing equivalent results. And then abemaciclib initially in second line with fulvestrant and ultimately in first line. So now we really have a large, robust body of data with all of these inhibitors.